Hello, and welcome to the 7th talk in the Mathematical and Computational Ophthalmology seminar series. My name's Paul Roberts, and I'm a Centre Fellow in the Centre for Systems Modelling and Quantitative Biomedicine at the University of Birmingham, where I work in the mathematical modelling of the eye. I'm the creator and host of this seminar series, together with David Crabb, who heads the Crabb Lab at City University of London, and Phil Luthert, an Emeritus Professor of Pathology at the UCL Institute of Ophthalmology. This seminar series aims to showcase research in mathematical and computational ophthalmology to the broader ophthalmological, vision science, medical, mathematical and computational communities, raising awareness of the exciting research being conducted in the field and encouraging future work in this area. Do subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. These are also listed on the worldwide page, the link to which can be found below in the About section. And our thanks go to Worldwide for instigating this platform for sharing scientific research. Before introducing our speaker, Giovanna Guidoboni, I'd like to remind our listeners that the deadline for submitting articles to the Investigative Ophthalmology and Visual Sciences, or IOVS, special issue on mathematical and computational ophthalmology is fast approaching, with a deadline of 1st of December. For this collection, guest edited by David Crabb, Phil Luthert and me, we welcome submissions of original research applying quantitative methods to examine any aspect of visual system structure or function. All submissions will be peer-reviewed and publication fees will apply to papers that are accepted. A link to the IOVS special issue advert can be found below in the About section of this YouTube channel. The term mathematical ophthalmology denotes the field which uses mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease, while computational ophthalmology is the field which uses computational algorithms, in particular AI, to preserve vision by identifying and predicting ocular features and risks. Thus, the focus of mathematical ophthalmology is upon elucidating mechanisms, while that of computational ophthalmology is upon prediction and identification. In our last seminar, focusing on computational ophthalmology, Piers Keane gave a fantastic talk on foundation models in ophthalmology, the recording for which can be found below on our YouTube playlist, together with recordings of all previous talks. This week, we're combining mathematical and computational ophthalmology with our speaker, Giovanna Guidoboni. If you'd like to ask Giovanna a question, then please do send these in via the YouTube chat, both during and after the talk. The talk will last for about 45 minutes, followed by 10 to 15 minutes for Q&A, in which I will relay your questions to Giovanna. So it gives me great pleasure to introduce Giovanna Guidoboni. Giovanna is the inaugural Dean of the Main College of Engineering and Computing, which bridges engineering and computing programs for the benefit of all students across the University of Maine system. She previously served as Associate Dean for Research and Professor in the College of Engineering at the University of Missouri, and was on the faculty of the University of Houston, Indiana University, Purdue University, Indianapolis, and the Université de Strasbourg. She is an elected member of the European Academy of Sciences and Arts and is founder and manager of G-Space LLC, which provides modelling and computational solutions for complex problems in engineering and life sciences. Giovanna also serves as co-founder and co-editor-in-chief of the journal Modelling and Artificial Intelligence in Ophthalmology. She holds a doctorate in mathematics and both a bachelor's and master's degree in materials engineering from the University of Ferrara. So I'll hand over now to Giovanna to present her talk, which is entitled Mathematical and Computational Modelling of Ocular Hemodynamics from Theory to Applications. Giovanna.
Thank you, Paul, for the nice introduction, and thank you for the opportunity to showcase the work that our group has been doing for the last decade in the field of mathematical and computational ophthalmology. So I have been very interested in fluid dynamics since I was in my studies in engineering. And so through my master thesis, my PhD thesis, and my postdoctoral work, I was looking at various aspects of fluid flowing in complex domains, for example, involving fluid structure interactions or free capillary surfaces. And I studied aspects related to the well-posedness, so existence, uniqueness, stability of solutions, as well as the development of numerical methods that would maintain at the discrete level the fundamental principles of physics that are embedded into the equations that we are solving. And also, I explored applications related to engineering technology, like coating flows, as well as the deformation of arterial walls, which is very important, for example, to understand aortic aneurysm. But my adventure in ophthalmology actually started in 2010 when I moved to Indianapolis, and I met Professor Alon Harris on the campus in Indianapolis. I was a faculty in the Department of Mathematical Sciences at IUPUI, and Alon was the director of clinical research at the Glick Eye Institute in Indianapolis. And so it was September 2010 when he invited several faculty from the School of Science to visit his clinic. And I was truly impressed by the many things that can be measured non-invasively in the eye of people just sitting in front of a machine without any further operation, rather than sitting and watching through something. So this is just an example of the various things that can be observed through the eye. And you can go from, as you can see here, the lipids that are in the tear film in the front of the eye, to the vasculature that is at the back of the eye, down to the capillary level, to the oxygenation of the blood in the arteries and veins, the 3D structure of all the tissues through the back of the eye, as well as the velocity profile in major arteries and veins in centimeters per second. So it is truly amazing to see that there is an organ in our body that can be imaged like that. I mean, if we think how much more difficult it is, for example, to measure things in the brain or in the heart, the eye really is there for us to see through. And of course, it is because it was meant for light to go through and enable us to see. And so we can take a pic. So as I was listening that time to Alon explaining everything that can be measured, I was thinking, if you can already have all this data, why do you need us? And actually, he was saying, yes, we have a lot of data, and the machines continue to advance. But the problem is that it is not possible with the tools that we have right now to actually combine this data together and understand what are the mechanisms that give rise to specific data, to specific measurements in every individual, and understand why, for example, certain people may have similar measurements with one machine, but one person may lose vision and the other one doesn't. So that is how my adventure in ophthalmology started. And in particular, I was intrigued by ocular hemodynamics because, as I told you, I've always liked things that flow. And actually, the blood flow in the eye plays a very important role in in ocular physiology and many of diseases of the eye. So I have organized the talk in this way. So I will first summarize some of the main challenges that we have been addressing, and then I will focus on glaucoma as a specific disease where I can give you a little bit of background. And I would like to show two particular examples in which our work in mathematical and computational ophthalmology has been helping in pushing the envelope of the clinical relevance of understanding the role of various risk factors. And then I would like to conclude with some examples of various activities that we have been promoting to really build an interdisciplinary world around science and medicine and technology, because it is only by working together across disciplines that we can really help everyone live a better life. So what are some of the main challenges that we are focusing? So as I told you earlier, so the eye really provides a window on many things, from tissue to blood flow to big vessels to most vessels to small vessels. But the fact is that the eye is inside the body, of course. So whatever we see in these measurements may be a consequence of factors that are local to the eye. For example, intraocular pressure, that is the pressure of the fluids inside the eye, the ocular shape. For example, if you are myopic or hyperopic, the eye may have different shape, different axial length, different lumen and diameter of retinal vessels, and so on. So factors that are local to the eye may influence the measurements that we acquire in the eye. However, as I said, the eye is connected to the body. So there are no local factors, meaning factors that are not only local to the eye, that influence what goes on in the eye. For example, there is blood flowing in the eye, but blood flows because there is a pressure difference between the pressure in the arteries and in the veins. And that pressure differential is actually established by our heart. So indeed, what happens in the eye is also affected by what happens in the cardiovascular system as a whole. In addition, the eye is connected through the optic nerve to the brain. The brain is another pressurized organ. So the cerebrospinal fluid pressure that actually is in the subarachnoid space around the optic nerve also influences what's going on in the eye. For example, also the respiratory function will affect the oxygenation that is in the blood, and that is the same blood that also comes to the eye. So already we have an interaction between local and non-local factors. In addition, there are differences in the eyes of men and women, of people of different ethnicities. Changes occur with age, and also with nutrition and environment, and so on. So the question is, when we then measure something in the eye, how do we know whether changes that we may see are due to factors that are local to the eye, factors that are systemic or non-local to the eye, or factors that depend on the individual? So that is really a mathematical or conceptual translation of the statement that I reported earlier from 2010. So yes, we can measure many things, but how can we know why we see what we see? And in particular, in this talk, I will focus on two factors: intraocular pressure and blood pressure. One is local and one is non-local. And I will show you how some of our work has helped disentangle the role of these two factors. So as Paul was mentioning at the introduction of the talk, mathematical modeling has really the goal of translating into mathematical equations principles of physics and physiology that basically embody the mechanisms driving certain phenomena. And so this schematic here on the right summarizes this concept. So I studied engineering, and actually I see a lot of similarities between engineering and medicine. For example, when we build a house, we have a structural component to it, like, for example, the walls, the roofs. We have hydraulics, we have pipes that carry different types of water, for example, hot water, cold water, wastewater, and I could continue. And then actually, if we look at the human body, also there is like the skeletal structure that helps with the structural component of the body. There are different pipes, arteries, and veins, and so on. And actually, many principles of physics that are behind engineering design can actually be used to understand several of the mechanisms in physiology that drive, in particular for this talk, the blood flow through the eye or the cardiovascular system. And mathematics can help basically translate these concepts into mathematical equations that then we can solve using computational methods and close the circle basically by comparing the model predictions with real data and understand whether the mechanisms that we put into the model in the first place are sufficient or adequate to understand what we see in the data. If actually the answer is yes, the model was good, we can capture what we see in the data, well, very likely it suggests that the mechanisms that we hypothesized at the beginning were the right ones. But it can also happen that what we actually see in the data is not what our model predicts. And that is informative too, because that would point to the fact that maybe there are other mechanisms that we did not deem as essential at the beginning of our model development, which indeed have to be taken into account. And so this is really an iterative process that can help really dig deeper into the understanding of what happens in the human body. And by the way, this synergy between mathematics, engineering, computing, and medicine is relevant in both ways. For example, there are many solutions that nature has already found, for example, self-healing processes that can be very helpful in engineering technology too. For example, self-healing asphalts, just to name one. So, I have written here on the right the rationale, the limitations, and some advantages of using mathematical and computational modeling to investigate mechanisms in physiology. And the I already talked to many of the advantages, and in particular, this concept of virtual laboratory, where we can use the simulations to look at the data and see whether we capture the right mechanisms. But also, I have to say there are disadvantages because every mathematical model is wrong. It is built on assumptions that are rough simplification of reality. But as the famous quotes go, all models are wrong, but some are useful. So our goal is to really develop models that are useful in identifying the mechanisms underlying the data. And so since that is the goal, we always have to think about the data. So as we develop the models, what is it? What are the inputs and what are the outputs that we will be able to either draw from the data or compare the data with? And so really, I see this modeling effort as complementary to data analytics, being that we have statistics, machine learning, or other methods of artificial intelligence, as I will show you an example at the end. Okay, so our group has been working since 2010 on various aspects of mathematical modeling of the eye. And this is a brief summary of the various aspects that actually called in much more than the fluid dynamics I was interested in. Just to give you an example, the intraocular pressure that establishes inside the eye is actually built up by a mechanism that has a lot to do with the movement of ions and charged particles across certain membranes. And that actually called for the utilization of mathematical models that are often used in semiconductors, as my friend and colleague Ricardo Sacco has taught me through the years. Even though fluid structure interactions in single vessels, as I was studying for the aorta, are very relevant also for the eye, for example, for the blood flow in the central retinal artery or in the ophthalmic artery, the microcirculation and the role that it plays in actually distributing the flow through the tissue in conditions with different blood pressure and intraocular pressure is extremely important. And so this calls for multi-scale models combining flow in big vessels, then through arterial, capillaries, venules, and back into the large veins. And not only blood is important, but of course what blood carries. And so it is oxygen and nutrient and how the circulation actually responds to that. Even though my expertise was more on the kind of theoretical mathematical side, by partnering with the high performance computing group in Strasbourg and now in Paris, we were then able to scale up the simulations and actually being able to capture 3D aspects of the blood flow and the biomechanics and scale it up to parametrizable parameters. So not only from the theoretical perspective, various methods were called in, but also from the application viewpoint, we looked at various diseases. So basically, the point is that when you focus on physiology, then physiological and not physiological changes affect many disease processes. And so today I will focus on glaucoma, which is the disease we focus the most on. But as a group, we have been working on several other conditions that I would be very happy to describe in a separate talk. So let us begin with glaucoma. So this is the second part of the talk. As I promised, I will begin with a little bit of background, and then I will actually go through two specific examples of how modeling helped understanding the disease process in glaucoma. So glaucoma is the leading cause of irreversible blindness worldwide. It is caused by the death of retinal ganglion cells that are the cells that are here at the back of the eye. They transform the light into an electrical signal that then is carried to the brain through the axon bundles that actually run through the optic nerve. The vision loss in glaucoma usually starts from the periphery, the only treatable risk factor for glaucoma is elevated intraocular pressure. So intraocular pressure, which I will often refer to by its acronym IOP, is the pressure that establishes in the fluid within the eye. And it can be decreased by non-surgical treatments like different types of eye drops, but also it can be decreased by surgery. The problem is that this treatment, so reducing IOP, is not always sufficient to stop the vision loss in glaucoma. Actually, it has been found that more than 25% of cases, actually, where the IOP is decreased, this treatment, however, does not halt progression. Also, over 1/3 of glaucoma cases do not really show elevated intraocular pressure. And by the way, when we say elevated, it means that implicitly we are assuming that we know what a good value is. But the truth is that what the optimal target IOP values are not really clear. They may vary among individuals, and such optimal values are not known. So yes, there is an approach to the disease, but it is pretty limited. So the question is, how can we help? How can we help prevent vision loss in that large part of the patient populations for which lowering intraocular pressure is not enough? So that glaucoma is a multifactorial disease is well known. And you can see here an incomplete list of the factors that contribute to the disease process. And among the most reported is blood pressure and also ocular hemodynamics. However, various clinical and population-based studies have reported results that are not unanimous. Low blood pressure along with elevated intraocular pressure has been found by many studies to be a problem, but other studies have also pointed out that high blood pressure may be a risk factor for glaucoma. So till actually it is understood why, when, and in whom blood pressure and which levels may be a problem, a therapy that aims at co-regulating blood pressure and intraocular pressure together is not viable. So our goals are, at least for this specific talk, to see how we can help address these two specific questions. So the one on the left, how do specific combinations of intraocular pressure and blood pressure affect ocular hemodynamics? So the underlying hypothesis is that if there isn't enough blood flow coming to the eye, or if there is too much, then the tissue will be damaged and cells will die. So understanding what is the impact of specific combinations of blood pressure and IOP in the eye through ocular hemodynamics may help explain some aspects of the disease process. The second question that you see on the right is, okay, now if we are able to then characterize this interaction in general, kind of in concept, how can we translate that into identifying subgroups of eyes, which means groups of people that are characterized by similar impact of IOP and blood pressure combination on ocular hemodynamics? And would these groups actually relate in a statistically significant manners to different ways of progressing in the disease? Of course, answering these questions basically means that we would be able to provide clinicians with an easy way to identify eyes for which just lowering IOP may not be enough. And in addition to that, giving specific mechanistic reasons as to why co-regulating blood pressure may be beneficial in those individuals, in addition to specific optimal quantitative targets for both blood pressure and IOP. So it is a very ambitious goal, but of course, if we succeed, or even if we go a few steps further into the understanding of the answers to these questions, we would have contributed significantly to the opportunities for maintaining vision in people. So when we started our endeavor in mathematical and computational modeling of blood flow in the eye, and I said it was 2010, the state of the art was as it is summarized here. So there was a lot of advanced work on the biomechanics of the eye, down to finite element simulations of stresses and strains in the back of the eye on real geometries extracted from images. However, there was no blood flow or no oxygen transport in it. So these were structures that were deforming, but there was no fluid. On the other hand, there were papers published with blood flow through realistic geometries of the vasculature in the eyes, also extracted from images, but these vessels were rigid. And so if a vessel is rigid, if a tube is rigid, it means that you can push as much as you want from the outside, but it will not deform. But as I said, what we are really interested in is understanding the relative role of blood pressure and intraocular pressure in the blood flow in the eye. And the intraocular pressure acts from the outside, squeezing the vessels. So if the vessels are rigid, we just cannot see that effect. Also, even though I will not talk about it in this talk, there were works published in the oxygenation through the retinal tissue. But again, a model or an attempt of modeling biomechanics, hemodynamics, and oxygenation together in a single model was not present. So that is where we started. And the theoretical challenges were many. For the people in the field, the fluid structure interactions are a challenge. They bring up, for example, partial differential equations of mixed type, for which the theory is not completely understood. These problems also bring multi-scale, multi-physics aspects that are challenging from both the theoretical and computational viewpoint. You cannot really reasonably model everything with 3D, so you will have some kind of simplified models calling for ordinary differential equations and other models calling for partial differential equations, and their interfacial coupling is also still matter of investigation in mathematics and scientific computing. And then finally, as I said earlier, we should remember that we should get back to the real data, and the real data are always much more messy than what we typically put in our models. So how do we do this comparison? So I decided to choose two particular models because they contributed to the field in different ways. And yet, in my opinion, they are both very important and call for really a diverse collection of expertise to work together and really be focused on what is the clinical need, what is the clinical relevance, so that we can see whether tools in mathematics and computing exist already, or if we need to develop them to actually solve the questions that are relevant. So let us begin with the lamina perfusion. So the lamina cribrosa is a tissue that is here at the back of the eye. It is kind of the plug of the optic nerve into the eye. And it is a collagenous structure that has several functions. So it helps maintain the pressure difference between the pressure inside the eye and the pressure in the optic nerve. It also allows for the nerve fibers, so the axon bundles, to actually pass through and connect the eye with the brain. And also the central retinal artery and the central retinal vein have to go through in order to nourish the retina. So the lamina cribrosa is considered to be a major site of injury in glaucoma. And one of the conjectured hypotheses is what is typically referred to as the biomechanical hypothesis. So here you see on the right kind of a rough simplification of this tissue as a circular plate. At the outer boundaries, this plate is actually connected with the sclera, that is the white shell of the eye. And as intraocular pressure increases, so the eye kind of inflates, generating a tension on the outskirts of the tissue. Also, as intraocular pressure increases, the lamina will deform more. Of course, it depends on what is the pressure on the other side. Here it is labeled as retrolaminar tissue pressure, which is connected to the cerebrospinal fluid pressure and the intracranial pressure in the brain. So it is a delicate balance of pressures that ultimately will determine how this lamina will deform. And now, if you go back to the picture on the left, if you deform this plate, then the axon bundles in particular that run through it will fill this deformation with possibly the collagen beams kind of doing weird things with the axon bundles, ultimately stopping or halting the signal processing to them and the axon transport through them and causing the cells then to die and the vision to be lost. So this is the biomechanical hypothesis of how retinal ganglion cells may be lost in glaucoma, again, due to this imbalance of pressures leading to abnormal deformations that then affect the axon bundles. However, this is a live tissue, so we know that to live it needs blood and or the nutrients. So interestingly, the vascular anatomy of this tissue is such that the blood comes in from the outside through the circle of Zinn-Eller, and it is drained from the inside. So basically, the blood is collected through the central retinal vein. So it is absolutely reasonable to conjecture that as the intraocular pressure changes, so as the lamina deform in different ways, and or we have changes in blood pressure, then also the blood flow through the lamina, which we refer to as blood perfusion, may change. And you know very well that if cells, wherever they are, do not receive enough blood and oxygen, they will die. And so this is another mechanism for nerve damage and vision loss in glaucoma, which is related to hemodynamic changes. So rather than opting for one of the two, we were really asking the question, how do biomechanics and hemodynamics combine, and how do changes in deformation in hemodynamics that are related to IOP changes and blood pressure changes through the lamina, how would that affect the life of the cells and possibly lead to nerve damage? So in order to model this, we then decided to look at deformable porous media models. So basically, these are models that have been around for centuries that describe the flow of a fluid through a porous structure, and the structure is deformable, which is, so you can imagine having the blood going through the vessels that are within the extracellular matrix and the collagen beams. And the end, we want to describe this complex structure as a kind of a continuum body with special characteristics. So these equations per se, as I said, are not new. They have been used a lot, for example, in geosciences, where the fluids that you have going through the porous media may be water or petroleum, and the porous material may be rocks and different types of sediments. And so what is really amazing about mathematics and that conceptual schematic that I drew earlier is that when you actually think in terms of the mathematical representation of these physical principles, you can actually take an incredible knowledge that has been developed, for example, in the field of geosciences, and apply it to study how people can lose vision because of glaucoma. At the same time, though, if we are really interested in having outcomes that are of clinical relevance, we really need to question whether, how, and to what extent existing models are adequate for what we need to study. What I mean in particular is that from the schematic on the right that I've been describing so far, I think that it appears clear that what drives the motion, being that deformation of the lamina or the blood flow through the lamina, is what happens at the boundaries of this plate. So in particular, we have pressures, different kinds of pressures and tensions on the outer boundary, as well as different blood pressures, so arterial and venous side, that are driving the blood flow through the lamina. So it is extremely important that here the boundary conditions that you see listed here are not homogeneous. So I don't have time in this talk to go through all the details of this model, but I just want to point out a few things. Basically, we have capital T is the stress tensor, the total stress that actually combines the stress that is felt by the structural part of the lamina as well as the fluid. U is the displacement, and P is the blood pressure inside the capillaries through inside the vessel through the lamina. This is the typical linearly elastic material. You have here a delta with another term that actually embodies the viscoelasticity. So if you don't have this delta and all the boundary conditions are zero, then this is kind of the typical BO model. Viscoelasticity is important, as you will see later, also because our tissues are always viscoelastic. So there is some kind of delay and capabilities of absorbing shocks. And also the boundary conditions must be non-homogeneous, again, to accommodate for the driving forces that are typical of this problem. Okay, so this work actually began as a very theoretical work, understanding whether these modifications on the right that we did to the classical BO system, so non-homogeneous boundary conditions and the viscoelastic term, would actually lead to a well-posed problem, which in mathematical terms means that actually it makes sense to look for a solution to it because it actually exists. And actually, this investigation led to a pretty interesting observation. So a solution to the problem exists in different spaces, functional spaces, depending on the regularity that we assume on the data. So translated into practical terms, it means that the intraocular pressure, as you see here in a real tracing from telemetry on primates, varies in time very quickly. So it is, and that is just physiological due to, for example, blinking, rubbing the eyes. And so when in mathematics, we say that our given data are continuous, are differentiable, so we need to give some regularity. Well, we need to take into consideration that in the real world, this may not be as regular. And so bottom line, what we found through the analysis is that only if the system is actually viscoelastic, we can have solutions that are regular in time. So basically, the viscoelasticity helps absorbing some of these oscillations. And so why should that be relevant for glaucoma? Well, actually, this led to formulating a new conjecture as to why we observe these hemorrhages in glaucoma patients. Basically, with age or with disease, the tissue may lose structural viscoelasticity. And so if you combine that, so the tissue being not as viscoelastic as it should be, and the fact that there are just physiological variations in the load, then this actually may lead to increased velocities because of the squeezing too sharp of blood vessels in certain regions of the lamina, which then may lead to ischemic damage and microstructural damage. So this new conjecture, again, that stems from a very theoretical work of well-posedness of partial differential equations of mixed type, has not been proved or disproved yet. So if anybody is interested in investigating this further, we would be very happy to work together. However, there are experimental clues that actually this may not be as absurd as we may have thought at the beginning. Actually, here you see some data on glaucoma eyes compared to normal eyes. And basically here you see different relaxation times of the stress, which is an indication that actually early glaucoma eyes may be less viscoelastic than normal eyes. So this concludes the first example that I showed you to basically share our really very eye-opening experience of how a very theoretical work focused on existence and uniqueness and so well-posedness of partial differential equations, when driven by putting into the model what was needed to understand the problem clinically, led to advances from the point of view of the theory of partial differential equations, and also the formulation of new conjectures on the disease process. So now I move to the second example that is related to the retinal circulation. The mathematics is much simpler, but the impact is equally strong, and I would like to share this with you. So now we move to a different part of the eye. The first example was on the lamina cribrosa, that is this tissue here, a little bit deeper in the optic nerve, as I said. The retinal circulation is really the one that is more directly visible because it is lining the back of the eye. And it is interesting because it has a single input, which is the central retinal artery, and a single output that is the central retinal vein. And they run parallel to each other for a tract in the optic nerve head. And here you see some of the imaging techniques that are available for different portions of the retinal circulation. So our goal is to try to understand and quantify how changes in blood pressure and intraocular pressure may affect the circulation in the retina. Why is that relevant for glaucoma? I told you that the various cells, including the retinal ganglion cells, have their soma on the retina, and then the axons that go down the optic nerve. And so also impairment in the retinal circulation may drive the cell's death. So here you see on the left an electrical circuit, and yet I am telling you that this is a model for the retinal circulation. And so that is one of the other wonders of mathematics. So basically the capability of taking now models that are used to describe the flow of an electrical current in a network to actually describe the flow of a fluid in the vasculature. So I have a few slides that just show the concept of this analogy. Basically, it is built on the fact that electric current is corresponds to volumetric flow rate, charge corresponds to volume, and electric potential corresponds to pressure. And here you can see on the left some resistances and capacitances. So the resistances that obey Ohm's law, if they are linear, then basically model an electric element where the difference in electric potential or voltage is proportional through the resistance to the electric current. And if we consider the flow through of a Newtonian viscous fluid through a cylindrical pipe, then we obtain the Poiseuille law that you see mathematically is identical to Ohm's law, assuming that we consider the pressures analogous to the electric potentials, the volumetric flow rate analogous to the current. And then here we go, we have an expression for the resistance that is related to the fluid viscosity, the length of the segment, and the radius. When the vessels are deformable, like we have in our case of blood vessels, then it means that the radius changes, so the resistance changes. And also we can accumulate volume, which is the analogous of charge. And so we have a capacitor. And so that is how the model is built. I want to point out that there are some arrows here on the arterioles and the venules, and basically they embody the fact that arteries and veins behave differently under a transmural pressure difference. In particular, the arteries are stronger, they have a thicker wall and a higher pressure inside, whereas the veins tend to collapse because they are thinner and the pressure inside is smaller. So actually this model is highly nonlinear, especially for the behavior in the veins. So I will not go into the details of the model predictions per se, because I think that the most interesting part is how do we use the predictions of the model? How do we use such a model to actually answer one of the questions that I posed at the beginning, which is identifying subgroups of eyes that are similar in the way in which combinations of blood pressure and diopy impact the hemodynamics. And so in order to do that, we will actually combine the mechanistic model that I showed you here with machine learning. And in particular, we will use for each individual the values of blood pressure and diopy to predict the blood flow and the distribution of pressures and volumes through the system. So now let us get to, as I said, the clinical application of this. So intraocular pressure and blood pressure are measurements that are very easy to do. If we could connect those measurements with how we progress in the disease, say 4 years down the road, that would be amazing, because these measurements are really widespread in every clinic. So the work that I will present now is related to a particular clinical study, the Indianapolis glaucoma progression study, where 115 patients were observed over a 7-year period. And if we just look at how blood pressure and IOP were distributed among these individuals at the first visit, basically you see a single blob. And actually, if you represent this in 3D with the histograms, you see that it is kind of a normal distribution. So if we only look at the values of blood pressure and IOP, it is not obvious that we can actually identify a group of people for which blood pressure and IOP have a different impact. However, if for every individual we take the values of blood pressure and IOP, run the model, use the model to estimate the distributions of pressures, flow rates, and volumes across the system, and put it back into our data set, now we have what we call physiology enhanced data set. So for each eye, we would have not only the measured blood pressure and IOP, but also the model estimated values of blood flow, pressure, and resistance, which is a way to put quantitative metrics of ocular hemodynamics back into the real data set. So why am I claiming that if we do that, we may be able to see groups of eyes that we were not able to see before? Because, as I said, the model is so nonlinear, and the veins tend to collapse when the transmural pressure difference is such that the pressure outside is higher than the one outside, that actually when you look at how the resistance in the venous or the resistance in the central retinal vein are distributed with respect to the belt that describes the distribution of blood pressure and IOP, you see that they are kind of pulled in different directions, thereby giving some chances that in the higher dimensional space captured by the physiology enhanced data set, we may actually be able to identify clusters. And so these are the results of clustering using fuzzy C-mean. I just want to show one. Of course, we used several techniques, including hierarchical methods, possibilistic fuzzy C-mean, and so on. And we looked at the clustering validity measures to see how many clusters. But basically, these are, you can see on the right, a visualization through TSNE of clusters that were stable in the data set and that are actually separable. And if we now map those into the two-dimensional space of blood pressure and IOP, we actually see that what seemed kind of just a single blob earlier, it actually embodies 3 different clusters. So this is interesting, but we cannot stop here, because these clusters will be meaningful only if we can show that actually they are indicative of different ways of progressing in the disease. So the question is, if today I am, for example, in the red cluster, am I a higher risk of developing glaucoma or progressing in glaucoma than if I am in the green or in the blue cluster? So that is why the data from the Indianapolis glaucoma progression study were so useful, because as I mentioned earlier, patients were evaluated every 6 months for up to 7 years. And now I will show you the results of changes over 4 years. And so patients that were in cluster 1, in this green cluster at the first visit, basically did not experience any statistically significant difference in their structural or hemodynamic markers after 4 years. Here on the left, you have different devices and different parameters that were used to assess the change clinically. So basically, this says that eyes that were in the green cluster were relatively safe. On the contrary, those that in year 1 were in the red cluster, they saw a significant progression in the disease on all fronts, both structurally and hemodynamically, whereas those that are in cluster 3 only saw mild changes. So this is this is truly important because in addition to being able to identify groups of eyes that show different ways of progressing in the disease, we also identified a group, that is the green one, for which actually progression was almost none. So this is not conclusive, of course. We need to test this on more data sets, but it provides an actionable insight on how we could develop testable hypotheses for techniques, for therapies that co-regulate blood pressure and intraocular pressure. And what I don't have time to explain is actually what is it that is different hemodynamically speaking among the three clusters. But briefly, for example, the blue cluster, the eyes have higher susceptibility of venous collapse, whereas in the red cluster, it is actually the capability and the functionality of autoregulation that seem to be limited. But again, this has to be validated on more studies. But the good thing, it is a testable hypothesis that could be easily applied to other studies. And we have already begun to do so. This is an example of the results on the Thessaloniki eye study. This is a much bigger study, including both healthy eyes and glaucoma eyes. And by doing the clustering the way that I explained, we were able to identify groups in which healthy eyes and glaucoma eyes have significant different hemodynamics despite similar combinations of blood pressure and diopy. And so this guides us on how to look for the differences. So now we are at the end of the talk. So a lot of different expertise combined here. So what are we doing and how are we moving forward? I emphasize that it was a choice to focus on blood pressure and diopy to drive the models because it is our goal to try to make quality healthcare as broadly accessible as possible. And so the mathematical models could be the link between advanced imaging techniques and easy measurements. And also by doing so, we may able to really bring to the people a more personalized approach to glaucoma care. In the current directions, we are moving, for example, in looking at other ways to use the eyes as a window on the heart and on the brain. We are looking at the pharmacological mechanisms that drive the intraocular pressure production, as well as from the theoretical perspective, both scientific computing and the integration with AI and ML to be more effective in actually analyzing the data. Building a community and inspiring young people and people of all ages to actually try to understand life is and the processes that are going on in our body is one of our priorities. Currently, we have organized live webinars from our labs, working with sponges to simulate the tissues. And also there is currently a high school course here in a high school in Maine that is based on this project. This work also led to 3 books that collect the fundamentals of the multi-scale, multi-physics modeling that was necessary to develop these models. So drawing knowledge from mechanical engineering, from semiconductor engineering, which basically it is really the broader aspects of physics combined with biology, as well as a particular looks at ocular fluid dynamics, what is it that is available to image and to understand it. In order to create also a community that could provide a hub for scientific research that can explain the details and the reproducibility of the mathematical models while maintaining clinical relevance, we founded in 2014 a journal called Modeling and Artificial Intelligence in Ophthalmology, that accepts papers in both LaTeX and in Word, and every paper submitted there will be reviewed by an expert in the more theoretical domain or the more applied domain, so that both aspects will be valued. So please submit your papers and contribute to build this community. So thank you. I want to thank everyone who contributed to this work. And Alon Harris, he has been a partner since 2010, really introducing to us the wonders of the eye and the physiology behind it, as well as Holly's group. Many friends and colleagues through continents and bringing, again, expertise in mathematics, scientific computing, AI, including also science communication and education. A big thank you to all the students, graduate students, undergraduate students, also high school students in the past who have, and now in the courses, who are interested in contributing to the research, and also the funding that made this possible. And finally, I want to thank you all for your attention.
Well, thanks, Giovanna, for a really fantastic talk. That was beautifully clear, and really was a great introduction, not just to your own research, but to the field in general. So thanks. That was really great. We have so a question has come in. If you'd like to ask questions, please do just post those in the chat at the side, and then I'll relay those to Giovanna. So the first question that we have here is from Vladimir Zubkov, and in relation to the work that you were talking about on laminar perfusion, he asks, how did you determine the mechanical parameters for the equations?
So it was very difficult. And by the way, we did the best we could based on the papers that were published. And so one thing that I always tell also, my students is we need to be very clear when we write the papers, where and how we took or we calibrated the model parameters, understanding that as if better knowledge or better experiments become available, they may have to be revisited. So having said that, what we did together with the group of Alon, we looked through published papers, so we did not conduct experiments ourselves. And experiments included, for example, induced pressure deformations on enucleated eyes or tissues that were excised. And so basically measurements both with and without fluid in it. And then measurements of different quantities, like, for example, the volumetric deformation or the deflection. And actually what is interesting is that the reported values were actually different across papers. And so what we did was basically summarizing also the range of variations. And then we focused on those parameters that were more consistent in terms of values and order of magnitudes across papers. Having said that, so it is clear that the kind of model constitutive equation that we used was linear, whereas it is very likely that in our body, the materials are actually non-linear. So there could be a lot of improvement there.
Thanks, Giovanna. We've got a question here from Rodolfo Rupetto. He says, ciao, Giovanna. This is Rodolfo. Thanks for a very nice talk. I particularly liked the use of a model to help cluster experimental data. You mentioned the importance of retinal vein collapse. Does the model you used account for transient collapse of vessels related to IOP changes during the cardiac cycle? And do you think that this is an important effect?
Ciao, Rodolfo, and thank you for the question. Yes, indeed, that is a very important point because the diastolic part of the cardiac cycle is the weakest when the blood pressure inside the vessels is lower. And yeah, the intraocular pressure varies, but not that much. And so we actually see that the susceptibility to collapse is much stronger in the diastolic side than the systolic side. So when we cluster, we basically put these quantitative features, like, for example, the maximum or the minimum pressure, the maximum or minimum resistance, and within this higher dimensional space of features, which we then use to cluster. So the similarities within the clusters will also account for similarities either on the systolic side or the diastolic side. And actually what we have found that what happens on the venous side, especially on the diastolic part of the cardiac cycle, seems the most relevant. I also want to emphasize that glaucoma is a slow disease. And so when we talk about systole and diastole, everything happens within a second. But actually the disease occurs over several years. So for me, it reminds me a lot of the damage by fatigue in materials, when you have that kind of oscillations that then at the end can break the material, even though the stress that you are inducing is not that high. And so the conjecture is actually that maybe as blood pressure and IOP oscillate, either during the day or between day and night, and maybe there is an impairment either in the vascular regulation or in the structural parameters of the tissue, then maybe there is this little damage that accumulates over the years and then will result in vision loss. But again, this is our working hypothesis, and we are trying to prove it or disprove it. I mean, hypotheses are there to be tested.
Thanks. We've got another question here from Perry Zekel, and they ask, are the data and model publicly available?
Yes. Everything is published. I am very happy, if you want to contact us. I also would like to thank some of the collaborators out there who helped us find typos in some of our papers, which we gladly corrected and published errata. For as much as one tries to be careful, sometimes that happens. And so we are extremely open to work with anyone and sharing not only per se the models, but also the ins and outs of how we came to certain choices or limitations and how to improve on those.
Cool, that's wonderful. Well, I think we're coming to the end of our hour now, so I'll stop the questions there. So thanks, Giovanna, for doing a great job in answering those, and for a fantastic talk. So I'll just close by saying that further details of all future talks can be found on our worldwide page, the link to which can be found in the About section of this YouTube channel. And do also subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. So thanks again to Giovanna, thanks to you for listening, and see you next time on Mathematical and Computational Ophthalmology. Goodbye.